RHD, or Rh blood group D antigen, is a protein encoded by the RHD gene. It is located on the plasma membrane of erythrocytes, or red blood cells, which are found in tissues like blood, bone marrow, and salivary glands. Because the D antigen is a part of the Rh blood group, erythrocytes on which the D antigen is present are considered Rh positive, whereas erythrocytes on which the D antigen is absent are considered Rh negative. The RHD protein is an autoantigen or self-antigen, meaning that it originates from within the body. It is highly immunogenic or highly capable of producing an immune response, so its function as an antigen is to bind to antibodies, proteins that result from the body's reaction to foreign substances. However, more specifically, the RHD protein determines an important part of a person's blood type, which is the Rh factor. A person's blood type consists of one of the four main blood groups: A, B, AB, and O, and is then further specified as Rh positive or Rh negative. RHD is intracellular, which means that it never passes through the phospholipid bilayer of the plasma membrane in order to exit the cell, and instead stays stationed at the plasma membrane. This means that the plasma membrane is its main target site. The formation of an RHD protein begins in the nucleus of an erythrocyte, specifically in an autosome called chromosome 1. This chromosome consists of two strands of DNA, one of which is copied through transcription to produce mRNA. The mRNA then exits the nucleus to reach the ribosomes, which are along the rough endoplasmic reticulum represented by the side of the tape measure with quarters. Through translation, the mRNA binds amino acids into a peptide chain called a pre-pro hormone, represented by the pearl necklace. A signal sequence of amino acids directs the pre-pro hormone into the lumen of the endoplasmic reticulum. Here, enzymes chop off the signal sequence, creating an inactive prohormone. This prohormone leaves the endoplasmic reticulum and travels to the Golgi apparatus in a transport vesicle. The Golgi apparatus is responsible for packaging the enzymes and prohormone into a vesicle called an endosome. The enzymes chop the prohormone into one or more active peptides, shown by the long black and silver necklace, and additional peptide fragments, shown by the black beads. And the endosome embeds itself into the plasma membrane, allowing RHD to reach its final destination. One mutation that can occur in an RHD gene is a nucleotide deletion, which causes a frame shift and premature stop codon. In order to display the Rh positive phenotype, a person must inherit at least one RHD allele. Most often, when a person has an Rh negative phenotype, the entire RHD gene is deleted. In this case, however, a pseudogene, or non-functional section of a DNA sequence that resembles a functional gene, creates a frame shift, meaning that it changes the way the DNA sequence is read. Thus, a translation termination codon is formed. Because the majority of people have Rh positive blood, people with Rh negative blood can face serious issues when getting a blood transfusion. If Rh positive erythrocytes enter the bloodstream of someone who is Rh negative, the person's body will start to produce antibodies against the Rh factor. This can lead to an acute immune hemolytic reaction, in which the antibodies the person produces attack the transfused erythrocytes, causing them to hemolyze, or break open, and emit harmful substances. The symptoms can be milder, such as nausea, fever, chills, and chest and lower back pain, or stronger, such as damage so severe to the kidneys that dialysis is required. In fact, if the transfusion does not stop once this reaction begins, it may be deadly.